Okay, there we go. Okay, so the recording has started, and the first thing we need everybody to do is log into the Hoffman cluster. Okay, so once you get logged into the Hoffman cluster, you'll see something like this come up. And we're going to start running some commands immediately. Okay. Quick show of hands. Who has gotten themselves logged into the Hoffman cluster now? Okay, who's gotten their QRSH already going? All right, and who is currently still waiting for their QRSH to start? All right. So since I need everybody to have their QRSH going, and actually I think to save on time in a little bit, what I'm going to have everybody do also is open up a second tab or a second terminal window or a second terminal tab or whatever. Get a second login going to the Hoffman cluster because you'll need it for a little bit. And then do the same QRSH command there. So we're going to start 2 interactive sessions with the Hoffman cluster. All right, quick show of hands. Who has not gotten their two QRSHs started? Okay, I'll give it a few more minutes. I think the Hoffman cluster must be having a busy morning. All right, quick show of hands. Who's still waiting on their QRSH to start? Okay, So put up your hand if you don't have both QRSHs running. Otherwise, I'm going to continue on now. Okay. So for Lewis, you're successfully logged in. You're just waiting for, are you waiting for one QRSH or both?
I'm waiting for both. So I typed it in and it said I can't use colon.
It said you can't use what?
Look, I typed in the QRSH in my terminal,
Uh-huh.
but it wouldn't let me run it.
Okay. Are you sure you put in the command correctly?
I'll double check. And how do I set up the second one?
Are you on a Mac or a PC?
I'm on a Mac.
Okay, so you'll open up a second tab in your terminal. You'll log into the Hoffman cluster for that one. And then you'll run the exact same QRSH command in that second window, or in that second tab.
Okay. All right, I think the QRSH worked out. I'll just run the second window.
Okay, perfect. You can even copy paste it from the first.
Yeah. So once it says like my name at 6441, does that mean it's done?
Yeah, as soon as you get your command prompt back after QRSH and you see your username at, and then it's probably going to be N and some number, that's telling you you're on a compute node.
Okay, great. I'm just waiting for the second one.
Okay, perfect.
Okay.
All right, cool. So we can get this thing started. All right, so you should be logged into the Hoffman cluster now, and you should have 2 interactive sessions going. Remember, as I said before, it's important not to do your computing on the login nodes. So based on the feedback yesterday, I'm going to have us actually run through a very basic Data 2 pipeline to get people familiar with the functionality. And as we go through it, I can explain some aspects of what we're doing. All right, so the first thing we're going to do is, to get started, we're going to copy over the data for the course. And copying over the data for the course is going to be running this line up, is going to be running these 2 lines with the arrows on them. So while people are kind of getting used to the command line and getting used to the way that my commands work, I'll have these blue and gold arrows pointing to commands that you actually have to run. As we get into it later, especially once I get into the R portion, I'm going to have fewer of those arrows pointing to the lines, just because the lines start to become a lot more obvious what you have to run and not. All right, so this first command we're going to run is just a standard Linux copy command, and that's copying data from my Scratch folder over to your Scratch folder. That should only take like maybe at most a minute. The second command you're going to run is it's going to be unarchiving a bunch of files, and your output's going to be actually different from this. This was an older set of data I was going to use for the class. I actually changed around the data quite a bit. So with the new class data, you're actually getting a little computer program in there to help, and you're also going to be getting a different 16S database. So go ahead and run these. I will check back with you in about a minute and see how everything went. Oh, the other thing that's very important is if you do run into trouble or you start getting error messages, let me know as soon as you start seeing them. I will probably enable screen sharing for you so you can share your terminal session and we can try and figure out where the error might lie. Oh, okay. So the CD command is going to be CD and then $scratch. So this $scratch right here, where it's $ and then scratch in all caps, you're just going to CD into this folder. And on the command prompt, that's the fastest way to get into your scratch folder. All right, I have another request for a copy of the slide, so I just put that link again in the chat window. Okay, so question is, are we doing this in both windows? No, we are not. So in one window, we're going to do the copying, which is what we're running right now. The second terminal window, we're not actually going to be doing anything with just yet. That's going to become important in a few minutes when we're working in R. All right, so Sarah, I can actually see from your copy paste. I think that says wsll.tar.gz. Those are those L's or capital I's after WS?
They're L's.
Oh, okay.
It should be,
so
yeah.
They should be ones.
Ones, okay.
Yeah, this is workshop 11.
That makes sense. Okay, now we're good. Thank
Awesome.
you.
No problem. All right, is there anybody who has not completed the untar process? So is there anybody who hasn't successfully run both commands on here? All right. All right, Lewis, it looks like you're running into an issue, or are you just, are you getting an error message?
Yeah, I'm just getting like no such file. I'm checking.
Okay, people are getting no such file. Okay, are you spelling it ws11.tar.gz, or are you putting something besides 11 there? All right, anybody who's getting errors on these commands, please let me know what errors you're getting and what command you type to get the error. Okay, so for Lewis's question, /u/scratch/m/Lewis_u, that is not going to exist. So in this case, you're going to actually use exactly this line because you're copying something from my Scratch folder to your Scratch folder. And then for Yajing, okay, so you weren't actually even able to CD into, oh, okay. According to Yajing, you found your problem. So yeah, after this, you'll be replacing my Scratch folder with yours. So that means that basically after this slide, you're actually going to be you'll be replacing this first letter with the first letter of your username, and then this with your username. All right, is there anybody else who has not successfully been able to untar? All right, show of hands, who has not untarred their file yet? Okay, I don't see any hands, so it looks like everybody's gotten the file untarred, so I think we can keep going. Also very important, if you get an error message, feel free to unmute yourself and say that you have an error message. Don't wait for me to call out for error messages because sometimes I'll forget to do it. And it also will just extend the amount of time this takes by quite a bit. Again, I'm still working on the timing, so I think we can get all of the commands run today. And my plan right now is we get all the commands done today, and then tomorrow I can go over in detail what we did and help you understand kind of the life cycle of microbiome data. So again, if you get an error message on one of these commands, just feel free to shout it out in the group, and then also copy the command that generated the error into the chat, and also copy the error message into chat. That's probably the easiest way for me to sort of do remote tech support for you. Okay, so what we've done in terms of getting started is we've gone into the appropriate directory in our Hoffman folders, and we've copied over the data we'll need for the course. All right, and then the next thing we're going to do is we're going to install Bioconductor. So that's going to require 3 commands. The first command is going to be module load, and then r/ and then the version number, and we're going to load this specific version. So I know somebody emailed me yesterday about a bunch of versioning issues. And as I mentioned before, there's kind of I mentioned yesterday something about dependency hell, and this is what we're trying to navigate around as easily as possible. So dependency hell is very annoying. And unfortunately, just about every microbiome package I've seen out there has some amount of annoying dependency issues, especially when they start trying to combine multiple programs. All right, and if I remember correctly, this process just took me a minute or two. So module load should be just about instant. Running R, I sincerely hope nobody made mistakes with this command because it's literally just a capital R. And then the last command we're going to do is install.packages and this bioc manager. So this is going to install Bioconductor. This one can take a minute or two to run. It'll ask you some questions about where to install it because it can install exactly where it wants to. So for that one, you're going to just basically agree to where it wants to install, which is going to be on your home folder. All right, show of hands, who has not finished installing Bioconductor? All right, so I think everybody's installed Bioconductor, going once, going twice. Okay, cool. I'm going to assume everybody's gotten Bioconductor installed, or they're, oh, yes? I hear somebody chiming in.
I have a question. It asked if I wanted to install packages, so I said yes, and then it said, please select a crayon mirror for use in this session.
Oh, yeah, okay. That'll come up. So we're going to pick,
it gives me like a bunch of different countries.
yeah, so we're going to pick 57, which is the closest one. It's in Oregon.
Oh, cool. Great. I didn't realize that that was the next step. Okay.
Yep.
Thank you.
Yeah, so we'll do that. This, again, this is going to take just a few minute or two to do. So yeah, we're picking Oregon. That's basically the I think UCLA used to host it, but I guess they haven't been hosting recently. But UCLA used to host a copy, which is the one I used to use. But since this hasn't been showing up for UCLA recently, I started picking Oregon. And so I'll wait a minute for everybody to get theirs installed. But usually this installation finishes in like under 3 minutes. Also, fun fact: if you order up your copy from Oregon, it's actually delivered by a hipster on an e-bike from Portland. Warning message: BioManager is not, so yes, BioManager is not available. And the reason for that is you made a typo. So it should be BioCManager. All right, so I'll give Yajing a minute or two to get that sorted out, but yeah, that should be easy to fix. You just remember also that the up arrow works in R. So if you typed in a command and that command had a little bit of a mistake in it, or you're going to type in a command that's extremely similar to it or whatever, if you hit the up arrow, you'll get back your previous command in an editable form. All right, looks like Yajing has it. All right, is there anybody who has not successfully installed Bioconductor yet? Okay, so the next thing we're going to do, so Bioconductor is sort of a package manager, and it just helps manage, so as I mentioned before, dependencies are a huge problem in microbiome software, and you're going to hear me say that a lot because dependencies are a huge problem in microbiome software. So Bioconductor manages these dependencies kind of behind the scenes for you. And so, for example, what we're about to do next is we're going to install Data 2 through Bioconductor. And it turns out Data 2 has just this unbelievably dense forest of packages that it depends on. A lot of these packages are like version specific and all that other stuff. So what Bioconductor is actually going to do is it's going to behind the scenes just know what needs to be installed and take care of installing it for you. So the good thing is you don't have to interact with it too much. You don't have to like wade through a big mess of installations that you have to do manually. That being said, the thing we're about to do is going to take a little bit of time. So the next command is going to be this one. It's going to be BioCManager and then 2 colons and then install. And we're going to tell it specifically to install Data 2 version 3.10. And once you have that going, this process is not fast. So I'm actually going to end my share for just a moment. And I'm going to share a different screen really quickly. Actually, is there anybody who didn't start the installation yet? Okay. Quick show of hands, who's still waiting for their installation to go? Or who's still waiting on their installation? It should be running. There's probably a lot of stuff going across your screen. Okay, yeah, I expect to see everybody's hand up. This installation can actually take like 20 minutes sometimes, depending on how fast the internet connection's going. Okay, so I don't see any messages from people indicating that they, yeah, I don't see any messages from people indicating that they are, not getting the installation running. So everybody's installation should be running. This installation takes a little while to run. So what I'm going to do now is I'm actually going to turn it over to Eloy, who's going to take attendance. And then after that, I'm actually going to switch to a different slide set really quickly so that I can go over something we're about to do. So I'll just turn it over to Eloy for a bit.
Hi, so can everybody see me? All right, cool. All right, everybody, same drill as yesterday. I'm going to share the link onto our chat room, and same thing as yesterday. I'm going to have you raise your hand after, lower your hand right now, but raise your hands after you complete the form. Okay? So let's see. Okay, the link is now on the chat room, so just go ahead and I'll fill it out. All right, good. So let's see everybody's missing. All right, thank you, Karen. All right, I sent a couple messages to those who didn't raise your hand, so please just go ahead and just fill it out before, and we're all set, okay? All right. Looks like everybody. Okay. All right, Michael, it's all set.
Cool, thank you.
Yeah.
All right. So if everybody can lower their hands really quickly. Okay, perfect. Next thing is, if your installation of Data 2 has completed, raise your hand. I don't think I'm going to be seeing any hands right now. Okay, that's what I thought. All right, so what I'm going to have everybody do is, as your installation of Data 2 completes, just put up your hand And when I see that everybody's installation of Data 2 is complete, I will keep going with that section. In the meantime, I'm going to switch presentations really quickly. There we go. Sorry, I just have to, every time I share my screen, I have to like reconfigure how everything's laid out. Okay, so while Data 2 is installing, which again is going to take a couple minutes, I'm just going to go over this part really quickly and talk about some of the stuff we're about to do. So for 16S sequencing, this is a kind of a schematic of the 16S region. And the region that we're specifically going to be sequencing on 16S, so these are the data that I have for you, is here. So we're actually sequencing the entire V3 and V4 regions together. This again is why I mentioned that the MiSeq winds up being important, is the MiSeq is the only Illumina platform that can generate long enough sequences to cover this entire pair of segments. And our reads are going to look something like this. We're going to have a forward read that covers V3 and goes somewhat into V4. We're going to have a reverse read that starts before or that starts after V4 and goes back somewhat into the V3 region, or at least into the conserved region between them. And these reads are going to overlap in the middle somewhat. And our goal is to be able to have some region in the middle of overlap where we merge these paired reads into a single read at their overlap region. The problem we run into, though, is the further you read into a sequence, and so on the x-axis here, and this is not, this is actually a much shorter read than even the one we're dealing with, but what we see on the x-axis here is we see position in a read, and on the y-axis we see the quality. So these are the Phred scores, just like we learned about yesterday. And we see that as you go further and further into an Illumina read, the quality tends to drop somewhat. So again, this is our goal, and I'm just going to kind of zoom in a little bit on the region. And so we have these two overlapping reads. We have some area in the middle that we needed to overlap, and usually for overlap, you want to go for about 20 bases. I think you need a minimum of 10, so 20 gives you a good margin of safety. And one thing you can see here is that the overlaps are actually more than you need. So a lot of times you'll have an overlap range of about 60, which means you can move this actual overlap region around a little bit. And for Data 2 doesn't like bad sequence, and Data 2 will start throwing stuff out if you have bad sequence in there. So this is actually kind of an innovation that I came up with, and it's not quite officially part of the Data 2 pipeline yet, but it's actually going to be soon. I'm working with the author of Data 2 on this. So we have some flexibility in where the overlap happens. And actually what I'm going to show you, I'll refer to something that I call the official way for Data 2, which is a human actually has to look at the quality score plots and then turn those qualities, and then basically by eyeballing the quality score plot, guess where the optimal places are to trim. You're going to see that I actually came up with a better way of doing it than that, and it's even fully automated. So how are we going to remove the human interaction there? And there's a couple reasons for this. So number one, a human looking at the same data on different days is probably going to pick slightly different trimming parameters. Somebody also has to figure out how many errors to allow in the read, and that's something that humans just aren't really good at all. So I'll talk a little bit more about that later, but it uses a metric for filtering errors that people don't intuitively understand very well. And the metric that it uses is called expected error accumulation. So expected error accumulation is a little bit different than what a lot of people are used to for error analysis. So I'm going to go back up a little bit. So if you look at this plot at this quality score plot, you can kind of tell where this thing drops into, say, quality score 20 or below. So we see here that it's dropped into quality score 20 by about the 40th base. And that's easy for most people to look at this graph and tell intuitively. But if you remember from yesterday when I talked about what the quality scores mean, at quality score 10, the base, so at quality score 0, you have 0% confidence, or essentially the machine is saying that it doesn't even know what to call there. At quality score 10, the machine is saying 90% confidence, or it thinks there's a 10% chance it made an error. At quality score 20, the machine is saying 99% confidence, or 1% chance of error, and so on and so on. So just as an example, if I'm looking at a position in the read and I have a quality score of 10, so in other words, 10% chance of error, then my expected error is 1/10 of an expected error. If I have 10 bases in a row where their average quality score is 10, then at that point I'm looking at, I have one expected error. So I can say that in this group of 10 bases with an average quality score of 10, statistically speaking, I expect there to be one error in there. Does that kind of make sense to everyone? So does that, for anybody out there, does that seem kind of confusing? Okay, I'm not seeing any responses, so okay, cool. It seems like people are saying it's clear. Yeah, so when I explain it that way, people kind of understand the metric that Data2 uses. So it's basically going to cut all the reads to the same length. It's going to look at the qualities for the read, and it's actually just going to take any read pair one of the members has over the allowable number of errors expected, and it's going to throw out that read pair. So one of the other complaints people have had with Data2 for a while is people don't quite understand that expected error metric, which actually kind of is sad because it's a much better way of filtering for read quality than just saying cut off where it dips below a certain point, because that takes into account the whole read. So if you have a whole read that's just kind of meh, it'll throw it out. Whereas if you have a read that just has one small area with low quality, it won't necessarily throw it out. So the problem is that typically quality scores are plotted this way. And if we look at the quality scores plotted this way, I can't tell you at what point I have one expected error here, because that actually becomes a matter of trying to calculate, looking at this curve and trying to calculate the area under the curve for the kind of error probability. And that is something that people don't do intuitively at all, because that's basically doing a ton of mental calculus on this thing. So it becomes a little bit easier to understand when you plot it out this way. And so what we see here is a black line and a dotted red line. The black line is looking at a subset of the reads from the sample and calculating the expected errors on those. So this one is actually showing the calculated expected error over the length of the read. And we see here is, as I mentioned before, Illumina reads tend to get worse as you go along the length of the read. And in this case, what I'm looking at is at the 83rd percentile. So this isn't taking an average, this is actually taking an 83rd percentile. So this is looking at reads that are one standard deviation worse than the average. And this black line, we see it go up kind of following this exponential pattern. on the reverse reads, we see that there's a much stronger inflection here, and that's because reverse reads tend to be lower quality than forward reads. And what I actually figured out is reverse reads, they tend to accumulate errors faster, but especially once you get a little bit further into the read, their rate really starts skyrocketing quickly. So if we look at these forward reads, we can kind of tell at any position of the read how many errors we can expect for reads that are 1 standard deviation worse than the average. Okay, and I have a question from Timothy Moore. Is this the expected error over the entire read, or is this based on certain window size? So again, first thing I'll point out, this is the 83rd percentile, so this is 1 standard deviation worse than the average. If I had this at the 50th percentile, I would be actually measuring the average read. But what we're looking at here is actually accumulated. So what I can say is for reads that are 1 standard deviation worse than the average, I expect that by about base 150, it's accumulated 1 error over the length of the read. By base 250, I can expect that it's accumulated about 3 errors over the length of the entire read. By base 300, I can expect that it's accumulated about 6 errors over the length of the entire read. So towards the end, it starts accumulating errors very quickly. All right, and I have a question from Sarah. Sorry to interrupt and go back to the Data 2 installation, but I'm getting errors.
And then I've gotten that for like a ton, like the R curl, genome info, DB, genomic ranges. So I've gotten a ton of those non-zero exit status.
Oh, okay. Has it continued running though?
No, it stopped. So it's back to the little caret guy.
Oh, okay. Has anybody else gotten that? Okay, so everybody else is still running. All right, Sarah, okay, so Timothy's also quit running. Is that correct?
I finished the Data 2 and I asked to update some packages, and so I updated those ones. And then it ran through all that and had a non-zero exit status on 3 packages. So I think it works. It might just be on the updated ones.
Oh, okay. And Sarah, what do you? And Sarah, is that what you got as well, or did you get something different?
I have it on like 8 packages that I had a non-zero exit status. Let's see. Yeah. And then I had errors above that says dependency short read is not available for package data 2. I had quite a few of those actually.
Okay, try running the installation again. So I think if you get one of those non-zero exit statuses, try starting the installation again. I'm wondering if we're actually causing some of these errors because we have like 16 people trying to install the same packages at the exact same time.
Gotcha.
Probably from the same distribution area. So just hit the up arrow and hit enter. The good thing is installed packages should stay installed. So you shouldn't, I don't think you'll lose progress as long as you had some packages that are successfully installed.
Okay. Thank you.
No problem. All right, and if anybody else runs into issues, first thing to do is try and restart your installation and then let me know if you still can't get it to work. The other thing that occasionally happens, although I think the Hoffman cluster fixed this, is occasionally you'll wind up on a node that actually has basically can't speak to the outside internet. I think they fixed those, but they used to have a problem where there'd be like always 2 or 3 nodes that can't talk to the outside internet, and you wouldn't realize it until you actually try and do something like this where it has to contact the outside. Although I don't think that's a possibility for anybody because if you installed Bioconductor, we already know that you have an internet connection. So I think probably the right fix here is just to try and restart the installation and see if that makes it go away. All right, so anyway, going back to what we're going to be doing, this is kind of helping you understand the pre-processing for the reads. this is something I can actually talk about very much in depth because this is a program that I wrote myself. So, and this was actually kind of how I became, this is kind of where I got my sort of expertise in Data 2 from, is I kind of learned Data 2 just trying to help people deal with issues related to proper trimming and proper read preparation. Because it turns out the Data 2 pipeline itself is great. This is kind of a pre-pipeline step, but it turns out this is the one where, and not surprisingly, this is the one that wasn't completely established. And this is the one where I was helping a ton of people who are having trouble. So one of the things you can see here is the black line again is the observed. The dotted red line is me trying to develop, it's basically my program called Figaro, trying to make a mathematical model for the error accumulation of your reads at the percentile of interest. And we see here that actually it winds up being pretty darn predictable. So I can use this line to predict at any given percentile what is the how many errors to expect from your reads. And the reason why I do this prediction is you can kind of see it better over here. The observed data is a little bit noisy, so you see a lot of up and down where it's just kind of twisting around the prediction line. And that's because to make this run faster, I don't run the analysis on every read. I actually take a subsample of your reads, sometimes even as low as like 1/200th of the reads. But since Illumina reads tend to be very constant read to read in terms of their error accumulation behavior, by building this model, I can smooth it out and get away with that. So when we run this program, we're going to see that it runs in under a minute. And then this kind of big confusing figure will help you really understand what's going on here. So the first thing I'll point you to is this y-axis over here, and also the x-axis. So the x-axis are read pairs, or I should say sites of trimming pairs, where if we trim at this position in the forward read and this position in the reverse read, we get our desired overlap length. So each of these is a possibility, and so every time we go forward in this direction, so if we go to 232, this will drop down to 268 because every time we add a base to the forward, we can take a base off the reverse. And the goal is to figure out the point where basically we can do our trimming, get our desired read lengths, but be tolerating the minimal amount of error on the reads. So if we look again at this y-axis, which is looking at the expected error accumulation over the length of the read, we see that looking at the forward read going in the forward direction, we see that it accumulates expected error values just going like this. So basically in kind of a stepwise way, because expected errors are always handled as integers, so we see it kind of increasing in this discrete manner. In the reverse direction, we actually see that it goes up much more quickly, and again, that's because I mentioned before, reverse reads are basically always worse quality than forward reads, so they tend to accumulate errors more quickly. This grayish silvery line along the top that basically hovers around 80. So this is our percentage of read retention. And this is basically saying, if I were to filter using basically these parameters, so for example, at this position, filter using 5 expected errors and 4 expected errors, I would retain, in this case, about 80% of the reads. And then the last thing we see here is this yellowish line, and this is the score that combination of trimming parameters gets. So the combination is a forward read trim site, a reverse read trim site, and a forward expected error and a reverse expected error. And you're going to see this combination show up a little bit later for something very important. And the score is, it starts off with the percentage of reads that are retained, because retaining reads is a very good thing here. And then it suffers a penalty for the number of expected errors that it has to allow. And that penalty increases exponentially for every error over 1 that it has to allow. So it can allow 1 expected error in the read with no penalty. 2 expected errors will get a small penalty. But for example, as it goes up to something crazy like here where it has to allow 11 expected errors, you see here that the score actually goes negative because, just in this case, for example, a read that's like 200, this is probably 280 bases, a read with 280 bases where you're expecting about 11 errors is probably a crappy read. So what we see here with this dotted line, this is actually where the program kind of zoomed into. And so what it did was it found this spot really quickly, and it said, if you trim right at this exact location, you can allow only 4 errors on each read. You can retain about 79% of your reads through the filter. And that was actually the maximum score that we were able to get. There are also a few other sites that were just about as good. So for example, if you look here, you'll see there was another site that probably could have been just as good where if we trim around 300 bases and around 200 and around 300 and 200, you would actually get a very similar score, and you would be able to trim there. You'd be able to retain about 80% of your reads. And in this case, you would have to allow one more error in the, one more error on the forward reads, but you'd be able to knock off one error on the reverse reads. So pretty similar. All right, this I won't go into too much. This is just saying what the program takes in and puts out. So as I showed you before, there's that plot of your errors, and it'll actually give you that as an output, so you can inspect that if you want to look at the quality of your reads. The main thing it needs to know is it needs a filtering percentile, or if you don't set that, it's going to default to 83. I generally just leave it at the default because I like the idea of just throwing out stuff that's one standard deviation worse than average. The things that it absolutely needs put in are, it needs the amplicon length, the primer lengths. The required overlap has a default value, so you don't absolutely have to put that in. And it needs a set of reads to operate on. So we're going to run that in a few minutes. All right, so I can terminate that one and go back to the normal slideshow. Okay, so for Lewis, yes, I will be sharing those slides. So those are going to go into the final version of the slides for the talk. I just didn't get to incorporate them yet because I was trying to kind of work out the timing on this one. So basically, this entire pipeline, the entire pipeline I made here, including navigating all the dependency issues, was actually done yesterday in response to your feedback. Because I got the impression that people are not familiar with this approach and wanted to kind of get an introduction to Data 2. So because of that, this was, because of that, I was making these last night, and that last set of slides will be incorporated into the slideshow that gets shared. Okay, so show of hands, whose installation is finished? And also I have a, okay, yeah. So actually when I did the installation, I think it'll even show it on here. Yeah, so you'll notice you got the same error I got. That was not actually a serious problem because everything actually worked. So I think everybody probably got the same message.
So I got that message too, but then I have warning messages below that. I don't know if those are bad news bears or not.
Yeah, I think this generates a lot of errors because Hoffman is kind of like, It doesn't always tell it where it can't write to until after it tries to write there. Okay, and it looks like Eva still has a download going. So I'm going to give people a few more minutes to get this going. And again, if you get weird error messages, it doesn't hurt to try and rerun the installation, because I think if you try and install something that's already installed, it'll just tell you that it's already installed, or it'll try and fix anything that looks like it's not right. So, all right, and it's actually 10:07, so we've been here for an hour anyway. So I think what I'm going to do is just to let everybody get it sorted out, let's pick back up again at 10:20. So that'll give about a 13-minute break so people can get their installations finished. You obviously don't need to sit there and babysit it the whole time. Good opportunity for people to take restroom breaks or get water, whatever else you need. I'm going to actually go grab myself some water. And as soon as I get back, I'll check for questions. So if you do have questions, let me know. Just leave them in the chat, and I will get to them as soon as I return.
Yeah, I mean, I have men's soccer, but the men's and women's tennis are kind of all that same thing. They do a lot of competition in the fall, essentially. Like, it's kind of more of a worse season in the fall than it is in the spring. So, like, we'll see how that kind of plays out for them. What the coaches want to do.
All right, for Sarah, this is where an in-person class works much better because I can go over and see your screen. So right now I can't look at your screen and figure out what the error messages are. Okay, let me let me get your screen sharing. So let me see. I think I have to make you a co-host. Try sharing your screen and tell me what if it doesn't let you, I'll make you a co-host.
Okay, yeah, it says host disabled attendee screen sharing.
Okay, try screen sharing again.
Okay. Let's see. Okay, I think I got it up.
All right, yeah, I can see it on there. Okay, try and run the try and run the installation again and let's just see what happens.
Okay.
All right, try typing quit and then open and close parentheses. And then actually don't save the image.
Okay.
All right, I want you to type in the word ping, P-I-N-G, and then space, and then 8.8.8.8.
Is that good?
Yep, and then just hit enter. Okay, you can hit Control C now. Okay, so what I was just doing was I was having you actually ping a server at Google really quickly, which is a pretty standard server for doing this, and you're just making sure that your node is connected to the internet.
Okay.
All right. Are you running in a Python virtual environment?
I could be, yeah.
All
I
right.
might have exited it last time I was using it.
Okay, try exiting the Python virtual environment. I think it might be, okay, yeah.
Okay. Oh no. Okay, so then should I just try doing the previous code again?
All right, try doing the module load command again for R. Cool, and now try running R again. So just R. Okay, and try hitting the up arrow. Okay, it doesn't have anything for the last one. That's fine. I wanted to make sure nothing was held over. Okay, so do you have the slides in front of you?
Yeah.
Okay, try typing the command to install BioC or Bioconductor.
Oh, small C.
Yeah, BioC Manager. All right, and then it's going to be, yeah, 57. Okay, and now try running the Data2 install. All right, so I think this is looking a little bit more promising because I'm pretty sure that last time it had failed by this point, right?
Yes, I think so.
Okay. Yeah, so that is a problem I have not run into before. But yeah, I've never tried to obviously do an install when I had a Python virtual environment going.
Gotcha.
So, okay, I think, yeah, that was a new one. Kind of.
Yes, this one looks more promising. Thanks so much.
No problem. All right, so I'll tell you what, let's actually even extend the break a little bit to 10:30 because Sarah's not going to be able to do anything until her installation completes. So I'm going to hang around here if anybody has any questions. And actually, Sarah probably, oh, and it looks like a few other people are still waiting as well. Yeah, this one can take a little while to install. And actually, Sarah ended up doing everybody a huge favor because I can pretty much guarantee that at least one or two people in the class would probably run into this problem at some point in the future. And we all actually, including myself, learned that, okay, so this thing doesn't work very well if you have a virtual environment activated. All right, so I'm going to give a few more minutes for everybody to get this thing up and running.
Hey, Michael, can you repeat the thing about the virtual environment? Sorry, you were muted.
Yeah. Okay, what was that?
Can you repeat what you said about the virtual environment?
Oh, sure. Just that if you're using a Python virtual environment, which is a pretty popular way of managing Python dependencies, you actually have the well, basically you have a bunch of dependencies now that I don't know what they are anymore. Like I kind of know what the typical Hoffman dependency that you have up and going, but when you try and do module load, it's basically putting in a bunch of dependencies that you didn't have before. And if you're in a Python virtual environment and you try and do module load, those two things are basically going to be fighting for control of your computing environment. And I don't know who wins, or probably they both don't. Probably to some extent they both lose a little bit. So in this case, what she was running into was she had a Python virtual environment activated, and I think that kept her from completely loading the appropriate dependencies for R. So it's kind of like some stuff worked fine, but some things it was trying to do, like accessing curl and that kind of thing, were apparently not working correctly. All right. And it looks like MinQ Lee, you're saying that yours wasn't done yet. Has yours completed now? All right. MinQ Lee, has yours completed? Because I know you just said it hadn't completed before. Okay, so I'm not getting many responses. Well, I said I'd wait till 10:30, so I'll wait till 10:30. Also, for people who are on here, one of the things that I do at Zymo is I also handle some of our like videos and educational stuff. So I put out a video, I guess a couple weeks ago, on understanding testing and understanding sort of statistics as they relate to testing and false positives. I'm getting ready to put out another video, and I was actually thinking, since everybody seemed to be pretty interested in the idea of the OTU approach versus the ASV approach. So remember yesterday when I was talking about how you deal with 16S sequences in the in sort of the context where you know there's going to be errors, and how you can either try and basically sharpen it or you can try and blur it to make the errors fall away. So that seemed like a topic that was of great interest to people, and I've noticed that a lot of people in the microbiome field, even the more experienced people, they don't completely understand how the OTU approach is defined. They frequently haven't even heard of the ASV approach, and a lot of people are really surprised when they learn about sort of these two approaches and how they're very different from one another. If I were to kind of turn that course material into a video for kind of wider distribution describing that, do you think would you be interested in that, and would you probably want to share it with your labs? Okay, cool. So I can take kind of that part of the lecture and probably spin it into a pair of videos. So okay, yeah. Yeah, I noticed yesterday there was kind of a big interest in that. So okay, so probably in the next week or two, I'm going to spin out a video specifically on that. In fact, I'll probably spin out 2 videos because I'm trying to do the approach where I make one video that's kind of a brief introduction to the topic, and then I make another one that's probably going to be like 5 to 10 minutes that actually goes more in depth. Because yeah, I think that's for 16S sequencing, that's becoming a critical area, and I think it's a critical area that a lot of people are just kind of coasting based off of what they've been doing, and they don't understand there's alternatives out there. Or worse yet, they start learning there's alternatives out there, but they only learn it when a journal reviewer starts bothering them about it. All right, so I think everybody should have Data 2 installed. The good news is that's kind of the longest, that's typically the longest single step anybody's gonna have to do for this course. All right, let me get my screen share up and going again. All right, let me reconfigure all this stuff on my screen. There we go. Okay, perfect. Okay, so as I mentioned, you probably got some of these little errors at the end. These ones didn't really cause me any trouble when I was testing it out, so I don't think they're going to be an issue for you. Okay, so the first thing we're going to do is we're going to set this variable path. show of hands from people out there. How many people are completely unfamiliar with R? Okay, so people have at least some familiarity with R. Okay, good. That's gonna actually make this go a lot easier. So I think everybody at this point probably then is at least familiar with this type of line where you're setting the value path and you're setting it to this string. Very important, use your own Scratch folder here. So in my case, it's the first letter of my username and then my username. In your case, the first letter of your username is probably going to be different, and your username is definitely going to be different. Also keep in mind, this is WS11, not WSLL. And the next thing you're going to do is you're going to list files in path. Yours are going to be slightly different from these. Specifically, these two files on the third line, or on the second line, you're going to see different files here, or actually you might see different, yeah, you'll see different files on, I think, in the second line. And this is just a sanity check. This is making sure that you have the right folder as path. The important thing is you should see a pair of files here called fecal and a pair of files called ZB standard, and there's going to be the forward and reverse reads in that. And these are files that you can actually hold on to. So these are actually just some generic files I use for testing a lot of different things. The fecal sample, that's basically just a fecal sample that was obviously for human gut microbiome. I don't even know whose microbiome it is. I think it's probably just a normal individual that we were using for testing. I actually wouldn't be surprised to find out that it was like an RA in a lab or something. And then the ZV standard, so that's actually the one I mentioned earlier. There's only really one Zymo Biomics. There's only one Zymo microbiome product that I'm specifically going to mention, and that is ZB standard, which is the Zymo community standard. And the reason for that is that's actually the data source for this one. So unlike a fecal sample, which is going to have a broad array of taxa in there, and it's just going to be a very complex microbial sample, the Zymo Biomics standard is 8 different bacteria, and they're all basically like reference strains. So that one's actually a super simple standard. The cool thing about it is it's known strains present in known relative abundances. So quick show of hands, who out there has done microbiome preps before? All right. Also, quick show of hands, who out there has heard of gram-negative biases or overrepresentation of gram-negative organisms in microbiome preps? Okay, yeah, that's actually a fairly significant problem out there with, you know, occasionally you can see clear biases in microbiome preps, and a lot of times you can, you actually wonder how reproducible the preps are. So one of my big projects has been, again, working on ways to improve microbiome reproducibility and standardization. And one of the big things that I tend to do is I have these standardized mock communities with known relative abundances and a mixture of tough and easy to lyse organisms. And if I run that through a microbiome prep pipeline and then I do the bioinformatics on it, which I've actually fully automated the bioinformatics now, I can get back reports telling me exactly how accurate it looks like it was. So I can see if there was biases relative to expected. All right, so we've listed the files. The next thing we're going to do is going to run these 2 commands. And this is a really good case for using the up arrow after you run the first one, because you'll notice that this is basically just doing the same thing for forward and reverse. So for the forwards, you're going to type this command. For the reverse, you only have to make a few changes. You change this to R's, and you change this to 2. Oh, and it looks like Yijing will be leaving us. So yeah, I will get the video of this sent over to Eloy as quickly as I can. Hopefully we can get it out today. I know that yesterday, it kind of comes down to how quickly my computer is able to process the video. So I think yesterday it took a couple hours. Hopefully today it doesn't. And Yijing, if I remember right, are you in Tantano's lab? Okay, well anyway, so these two things happen pretty much instantly. And then running these two commands should also be pretty much instant. And I have a question from Tim Moore. So it might help if the commands are sent in a text file. That might avoid typos for people. So yes, actually for my workshop too, I used to give people a text file with all the commands. Unfortunately, yeah. So unfortunately what I learned that way is if I give all the commands, the students do tend to be happier during the class because they all wind up copy pasting the commands. What I also learned is that the students wind up being kind of sadder about a week later because what people start getting in the habit of doing is blindly copy pasting commands, and they wind up not retaining as much. So definitely, if you get error messages, or if you make typos, or if you think you make typos that you can't figure out, or if you get error messages you don't understand, you know, say something or post in the chat window or whatever, because actually typos are an important way of learning how things behave and you know, how to basically identify typos. The best time to learn troubleshooting is typically in class where I can give you a hand. The worst time to learn troubleshooting is basically the week after class, where I may not be around to lend a hand, and you may just be kind of struggling. So I kind of expect students to learn from each other's errors. So I get your request. I usually try and avoid doing that because in the short run it's helpful. In the long run, it actually ends up costing people. All right, so Lewis has a question, and it looks like this is being caused by, yep. All right, so Lewis, what I need you to do is type in the word path into your R prompt and tell me what path comes back as. Okay, yeah, I was gonna say I noticed that typo. Yeah, and Eva's right. So you set them both to FNFs. And yeah, that's why you got nothing, is your FNFs was probably right with the first line, and with the second line, you set it to something that is actually putting nothing in there, because there's not going to be any file ending in 002. So it would return nothing. So try rerunning those lines, just fix, so the second line, you need to have it go to FNRs, and you need to have it be 001. So as you can probably guess from the command, what it's doing is it's looking in that folder for files ending in underscore R1 underscore, or in the second case, R2, and then underscore 001.fastq. All right, so Lewis, when you reran the commands, did you see what you expect? What was the expected? Okay. Cool. And then with regard to the value of, and so this is actually why I was saying that I tend not to give out the sheets, because one of the most valuable things in class is people learn how to troubleshoot, and people see how to troubleshoot if they're not troubleshooting themselves. So this was a good example where you've kind of seen where things can go wrong and how to recover from it. All right, so there's no command on here that we're going to run, in part because some of us may or may not have visualization terminals running to our instance. But the official method in Data 2 for for trying to figure out how to set quality profiles is looking at these plots of qualities. I'm going to be frank, this friggin' sucks. But this has been kind of the standard method when running Data 2 for doing the quality analysis. And what I was finding was people had a tendency to not be able to always pick the best trimming sites for the forward and reverse reads. And people tended to be very overly restrictive with the expected error rates. So what I saw was a lot of people were frequently running their trim and filter step, which is the next thing we're going to do, or yeah, it's going to be the next kind of process we do. And unfortunately, what a lot of people were doing was they were losing like 80% of their reads to the filtering process because they were not setting good values. All right, so you've already opened a new terminal and already gotten a new session going. So in that one, if you're not already there, CD into your Scratch folder and then CD into the Workshop 11 folder and type LS. And when you type LS, you should see that there's the data folder. And the data folder is where our sequencing and reference database live. And then there's the Figaro folder. And the Figaro folder, so Figaro is that program I was talking about earlier that will find the, that will actually find the trimming parameters for us. So you're going to type in module load and then Python 361. So this is a version of Python that Figaro likes running in. You'll see a little message probably saying that it's doing some stuff. And then the next thing you're going to type is this really long command. So you're saying Python 3, and you're telling Python 3 to run Figaro, and then the Figaro.py program. You're going to give it an amplicon length. If you're actually, so this line is unnecessarily long because I'm actually putting in the long version of the arguments. There's actually short versions of these arguments as well, but for the sake of this one, I want to use the long ones just so people can be very clear about what it is. So amplicon length is the longest amplicon we expect to generate, and I have a all right, so Ava, oh, you're typing in part of my command prompt there.
Oh, I totally am. Thanks.
No problem. Yeah, so CDWS11 should be what does the trick. All right, so as I said before, so things like the amplicon length, that's going to be the longest amplicon you expect to generate. So 16S sequences can have a little bit of variability in their lengths because they come from different species. Typically, they're not going to be varying by more than like a dozen or two bases in either direction. So this needs to be the longest sequence you expect to get from your amplicon, which is going to be 470. Forward primer length is 16. Reverse primer length is 24. So these three values are going to depend on the library prep method you use. So this just happens to be from the library prep method that was used to generate the data that you see that you have. If you use a different library prep method than the one that was used here, then your numbers may vary a little bit. Although, for example, this is V3, V4. And for a V3, V4 amplicon, you can typically expect about this size of amplicon. You know, it may go up to like 500, it may go down to 460, but you can pretty much expect the size of amplicon depending on where your primers lay. The input directory is the directory where the sequence is, and in this case, it's going to be data. And this program should run in about a minute or less. All right, so I'll give everybody about a minute or so to get this going. If you get an error message, let me know as soon as it happens, and I will see if I can get you sorted out. If the program works, it'll usually, it'll show you something that looks like this. You'll start seeing something that looks like this, although you may have to scroll up to get it. So I see there was an error message. All right, Python 3 command not found. I have a sneaking suspicion that you're getting that error message because you did not run the module load. So did you run the module load Python /3.6.1? Okay, try running the module load command and then just use the up arrow to rerun that same command and see if that sorts it out. So the primer lengths actually affect the primer lengths affect how this calculates the amplicon size. So the output is going to look like a bunch of lists of values. You're going to see something that looks a lot like this. All right. And from MinQ Lee, I am getting some hiccups on the last slide regarding character 0. So can you show me exactly what you're getting and what you're running? Okay, so I can already see what the error is. You have /m//Andrew MI. You actually should be /a/Andrew MI. You have a typo in your path. So fix your path value and then rerun those lines exactly as they already are, and you should get the correct values again. So, okay, so for Vivian, it looks like your CD $signs. Oh, you have a typo in Scratch. So it should be CD $Scratch. You made a typo there. So change that to Scratch, and then the CDWS11 should work. Okay, so I'm going to keep on going with this. All right, so when we ran it, we should get something that looks like this. You probably got a whole list of these guys that are giving you combinations of trimming positions, maximum expected errors, read retention, percentages, and scores. You probably have to scroll up a little bit to find the top value, but it's actually this first value that you got back. That's the one we want. It's the one that got the highest score. All right, so we're going to return back to our terminal that's running R. And we're now going to do some more, tricks to handle our sample names. So we're going to set this value as sample.names, and we're going to run this sapply thing. And basically, this is extracting the sample names from the file names. All right, and I think once you've done that, you should be able to see that you have fecal samples and you have zymobiomic standards. We're then going to go through this process. So this is setting up our file names for our filtered files, because our next step is going to be the trim and filter. So we're setting up names for our filtered forward files and our filtered reverse read files. And we're just going to set it up using this scheme right here. And then we're just going to check and make sure they make sense. All right, and Eva, you have a plus on your command line. I am not sure what, is that an R or is that on just the bash? In R? Okay, I'm not exactly sure what that plus is. Is it causing you a problem? Oh,
I
okay.
didn't finish the line of code, and it's just like, you know how in R you kind of get that plus, and I'm not sure how to get out of that here.
Oh, okay, I see. Try hitting Control C and see if that, yeah, or you can, and then
Yes,
try.
thank you. Perfect.
I'm sorry, I wasn't done typing sample names for the last.
Oh, okay. Did you, do you have a copy of the slides?
Oh, right, I could.
Okay, so I think we can go on from this one. So we've now set up file names for our filtered files. And then we're gonna do a little bit more file name management, which is basically just adding these to names. So just run this command really quickly. All right, for Vivian Enriquez, so it looks like you're in your Scratch folder. Type ls and tell me what you see there. Okay, I see what the problem is. All right, so type this command as I give it to you. Tar, T-A-R, space, dash, XVF, so that's gonna be V as in Victor. And then space, and then type WS and hit the tab key. And that should autocomplete to the file name. Once you've got the file name showing up, hit enter. Okay, perfect. Yeah, I figured that was it. So yeah, you hadn't unpacked the files yet, so it was not going to be able to handle them. All right, so more file name management should be, all right, so this should be fairly quick to do. All right, now we're going to actually run the trim and filter process. So right now, don't pay too much attention to the bottom block because we're actually using this for reference. We're not using this for We're not using this actually for not typing anything here. All right, so we have the command here, and actually in this case, don't copy paste from the PowerPoint because PowerPoints tend to change characters a little bit. But I put this here so that it's a little bit easier to read. So we're going to set this value as out, and we're going to run our filter and trim process. And so we're going to pass it our forward files, our filtered forward file names, our reverse files, our reverse filtered file names. So our trunk length, and you notice there's these two values here, which we can actually pull from this. Max n equals 0. So we're basically saying throw out any read that has an n value in it, which is fine because data2 can't really handle any read that has an n in it anyway. It'll just throw it out. Max EE, so that's the max expected errors. And we're setting that to 5 and 4, which are values we got from here based on the prediction of Figaro. Trunk Q, basically if you have a read that has a quality score of 2 at some point, you're just going to cut it off there. We generally won't run into those unless they're really bad reads. This is remove phi-x. So this is just going to remove some reference DNA that Illumina likes to use. Compress is going to tell it to compress. And actually for this multi-thread value, you see multi-thread equals false here, but actually we're going to run multi-thread equals true. That's going to make it run faster because we actually set up our instances with 2 cores instead of 1. So multi-threading should cut the time to run these in half. For this one, if you didn't run with multi-thread, that's fine. The trimming process actually runs fairly quickly even without multi-thread, but on the future operations, we're going to actually run multi-thread equals true. So I'm going to give a minute or two for this to happen. Could not find filter and trim. Oh, I think I know where that came from. You know what we forgot to do is actually, yeah, and I think that got cut off the top of this slide. Yeah, I know exactly what we forgot to do. We went through installing Data2 and we forgot to actually tell, we actually forgot to load it as a library. So I just sent out a command, try running that, and that should get everybody up and running.
Hey, Michael?
Yeah?
It says loading required package RCPP.
Yep. That's just what it does when it starts up. That's not an error message. That's just telling you that it's loading up a package. All right. So I think once, yeah, once you've loaded, once you've done the library command, you should be able to run that trim and filter.
Sorry, I'm getting an error.
What error are you getting? Oh. So you are actually missing, if you look between, yes, exactly. You're missing filt Rs, and that should go after FNRs. So yeah, you should be able to fix that by just hitting the up arrow and putting in that value, and everything should be fine at that point. No problem. All right, so let's keep going with this. All right, so this part of the process is going to take actually a few minutes. So we're going to run the learn errors process. Basically, as soon as this is done, this will save the error values. And I'm just going to jump ahead to the second slide. You're going to do the exact same thing for the reverse, only you're going to feed it the filtered reverse values. So on this one, you definitely want to use multi-thread equals true, because this is going to take a while. It'll take even longer if you don't. And so as soon as your error finding finishes, feel free to jump ahead to the reverse.
I'm having an error with the trim, or the filter in trim. It says that it's removed all reads.
That's definitely not good. Did you check and make sure that your FNFs and your filt Fs are what they were supposed to be, and that your Rs are also what they're supposed to be?
I guess I did filt FS, and those were good, and filt RS, and those were good.
And they're both linking to the appropriate files. They're both linking to
Yeah.
parent N2.
They're both linking to fecal R fit fast Q, and then like the R's and F's are corresponding.
Okay.
And when I did it for the other stuff, or for or FNRS and FNFS, it looked good.
Yeah, I'm not sure why you'd be getting that error here.
Should I try to like redo the names and do it again? Or like getting those files?
So
Yeah.
actually a better idea here might be type those values in. So type in FNFS and FS and the same thing for the R's, and then paste that into the chat window. And let's just make sure that you're referring to the right files here. Okay, so it looks about right to me. Did Figaro run correctly?
I'm sorry, what do you mean? Say that again?
When you ran Figaro to examine the files,
When did we?
so that was the one that generated the output that looks like this thing at the bottom of the slide.
Oh yeah, that ran correctly.
Okay, so it's very strange that it would be
Right. That was the other window, right?
Yeah, that was done in the other window.
Trim position 305.225. Yeah, that was all the same. Yeah.
Okay, so that's weird because Figaro is actually operating off the same set of reads here. Okay, so that tells me that the reads are probably okay. You don't have anything weird with your set of reads compared to everyone else. All right, when you ran the command, did you copy and paste it from the PowerPoint slides or did you type it out by hand?
I had typed it out by hand.
Okay.
But oh, you know what?
Oh, did you see something? What's up?
Yeah, I think I figured it out. I don't know,
okay.
but it's good now. I'm getting the same reads in and reads out.
Oh, okay. Okay, cool. So I think I can keep going then. Okay. Run the learn errors command as well. So run on the forward and reverse. Quick show of hands. Who's gotten their learn errors command for both forward and reverse completed? Okay, who's waiting for their learn errors commands to finish? Okay, so they're not quite done yet, so I'll give it a few more minutes. I'll tell you what, if everybody wants to take a quick restroom break, I'll pick up again at like 11:23 or so. By then, the process should be completed for everyone, but occasionally learn errors does take a few extra minutes.
I should be getting a little carrot back when it's done running those error forward and reverse, correct?
Yeah, the little arrow thing.
Yeah, thanks.
All right. 1 second, I have a question coming in I'm going to respond to real quick. All right, I think everybody should, I think everybody's learn errors process should be completed. So we should have errors on the forward and reverse. Here's an example of what you get since Hoffman isn't so good about letting us plot stuff easily to our screen. This is just looking at the probabilities of errors for bases at a given quality score. So we're seeing all the possible changes. So this is, for example, an A to A change, which obviously isn't really a change. So that's why we see that basically it's completely confident here. Here, as we have increasing quality scores, we have changes in our confidence. And what we're basically seeing is if it calls a very high quality score, it's probably a little bit overconfident. If it calls a low quality score, it's probably a little bit more accurate. All right, so now we're going to actually do the denoising process. And again, the two lines are going to be almost identical for the forwards and reverses. Just remember on this one, so this is this is generating our denoised sequences. We're running the, so DATA stands for Divisive Amplicon Denoising Algorithm. And we're going to give it our filtered forward reads. We're going to give it our error model, and we're going to tell it to multi-thread because this will make it go a lot faster. This one should run fairly quickly. This one usually runs in about 1 to 3 minutes. For our reverse reads, we're going to do the same thing, only in this case, we're storing them as the R's. We're going to give it the filtered reverse reads, and we're going to give it the reverse error model. So run these two. This should take probably no longer than 5 minutes. All right, so I think this part of the process should have finished. All right, so let's go on to this part. So we're going to look at the yeah, we can look at our data Fs, and this will kind of give us an idea for what we came up with. So the data Fs actually only found 414 sequences out of the 29,000 unique ones. So it basically said that it saw 29,000 different sequences in your reads, but based on the error modeling, it actually thinks there are only 414 original sequences in there. In the zymoviomix standard, it actually said there's only 138 sequences, which makes sense there'd be a lot less because, quite frankly, this is a very low complexity sample, so there's only a limited number of microbes. So the next thing we're going to do is we're going to try and merge read pairs. This process also runs fairly quickly, so we're going to save as mergers. We're going to run our merge pairs operation. And that's going to be giving it the data Fs and the filtered forwards, the data reverses and the filtered reverses. And again, like I said, this tends to run fairly quickly. This runs in about a minute or two. All right, so this should have run successfully for people. Like I said, this only takes usually a minute or two to finish. So if you do have a look at what you get out of there, you're going to get this. I think it off. Okay. Well, anyway, if you have a look at what that generated, you'll see that it basically generated a bunch of a table of sequences that were picked up. All right. So the next thing we're going to do is we're going to make our sequence table. We're just going to call it seq tab, and that's going to be making the sequence table off of the merged sequences. This runs pretty much immediately. So I'll give everybody a minute or two to get this going. All right, so I think this should have gotten completed. You'll basically get a table that looks like this. This is going to give you the size distribution of your sequences. And basically the sequences should all be very close to the expected amplicon size, although probably just a little bit shorter. So it's going to be it'll be close to the amplicon size minus the size of the primers. If you see anything that looks like it's much longer, much shorter than what's otherwise observed, it's probably garbage, and you should check it out to see if it's just a non-specific priming event or something like that. So I'll give everybody a minute on this. Okay, so once you've done that, we're going to remove chimeras. So something that is interesting about Data 2 is it can't identify chimeras because chimeras look just like real sequence. So chimeras have to be removed in a different way. The nice thing with the Data 2 process is, unlike the OTUs, which because you're doing a blur process, obviously they're going to be a little bit fuzzy, removing chimeric OTUs can be a little bit more involved. With Data 2, essentially the process it's going to do to remove those ASV chimeras is it's going to look at all the amplicon sequence variants that were detected, and it's going to look at them by the frequency with which they were detected. And that's because pretty much by the way they're made, a chimeric read is going to have two other reads that can explain it, or two other reads of origin where halfway, somewhere in the middle, you kind of see it transition from the first read to the second. And it's going to be less frequent than either of the sort of parent reads. So what Data 2 is going to do is it's going to look for any reads that are low abundance that can be explained by two higher abundance reads. So we're going to run that, and what we're going to see, we're going to run a couple of these things. So first we're going to run this process, so seqtab.noQIIME, and that's going to be the remove bimera de novo. So that's using a reference-free method. And what we see here, and this is actually a pretty typical pattern on 16S, is it identified 2,341, they call them bimeras, but usually they're just called chimeric reads, but it identified 2,341 of these out of 2,855. So what we have is we have a ton of chimeric reads, or a ton of chimeric sort of diversity. which isn't actually all that surprising, that's pretty typical, is chimeras tend to show up as low abundance reads that are very diverse. So that ends up leaving us with only 514 reads that are unique and we're not chimeric. And we see here that unfortunately it looks like we had a ton of chimera in our library. That sometimes happens. Sometimes you can optimize your library prep to get rid of that. And what we're going to do down here finally is we're going to generate that size distribution again, this time looking at our chimera free. And we see that especially a lot of these short reads were kind of cut away, but overall we lost stuff in every category, and we even lost our shortest group, our shortest read. Okay, so Soyun Kim wants to know what's the difference between if we use true or false for multi-thread. And yeah, the find errors is actually, I think, about the longest process that it's the longest process that Data 2 does. It's the most computationally intense. And that's because it's actually running a machine learning model. So essentially what it's doing when it's doing the find errors, it's using something like expectation maximization. Essentially what it's doing is it's running a bunch of iterations on the sequences, and it's trying a bunch of different hypotheses. So the first thing it's going to do is it's going to say, it's actually going to hypothesize that your entire sequencing experiment was done on a monoculture. And so basically with a monoculture, it's essentially hypothesizing that there's only a single 16S sequence present, and that every sequence that doesn't match it is generated by error. And what it's going to find really quickly is that that is not actually a reasonable hypothesis unless you were doing a monoculture. So then it's going to go out and say, okay, can this be explained maybe by, 2 organisms and 3 organisms? And it's just going to keep going until it figures out an error model that actually makes sense with the observed data. And that's when it stops. Because it's doing that, and because it's doing kind of a long iterative process, it's much better to let it run on multiple CPUs, which is what the true is doing. So in theory, in fact, what I've seen is you get basically no difference if you run a multi-threaded operation or if you run with multi-thread equals false. It's just a question of how long it takes you to get to the end. And so it took a long time. It took a long time with multi-threading, and we're multi-threading on 2 cores. If we hadn't done multi-threading, then it would have taken basically about double the time, which would have been kind of painful. All right, so we can also do tracking of our reads. And this is the operation for doing the tracking of our reads. Basically, you're going to do all these different commands, and what you're going to be able to generate is sort of this read tracker, table, which is basically saying in both samples, we started off with about 62,000 reads. After filtering, we were down to about 48,000 reads, which is pretty typical. You tend to lose a lot of reads during filtering. During denoising, we ended up losing a couple of reads. Denoising doesn't typically get rid of a lot of reads, though, in either direction. Merging is notorious for getting rid of reads if you have bad trim and filter parameters. So trimming parameters are very important. If you have bad trimming and filtering parameters, you'll actually see yourself lose a ton of reads during the merge. And then we ended up losing actually more than half our reads that made it to the merge because they turned out to be chimeric. So what this would actually tell me is I might want to go back and check on my library prep method because I might be PCRing too much, or I might have some other issue that's causing me to generate a lot of chimeras. And then the last couple things we're going to do, we're going to assign taxa. And for this, you have some taxonomy databases already in there, specifically the Silva ones. So for this one, actually, it will work if you keep it as my folder, because you have access to this data. You should have access to these data sets already. So my folder, I think these are publicly readable. Ideally, you want to use your own copy just because it might get slow if everybody's trying to read off of mine. That's going to probably jam up the hard drive. But if you actually don't change this, it probably will still work. And then you're going to run taxa.print. And you'll see that it generates this. All right, and then the last thing we're going to do is we can write out that taxa table for basically storage on the hard drive. And this is actually the last step in the process. So I'm going to hang around for a few minutes in case people have questions or let people get caught up. But once we've gotten through this, we've actually done a data to pipeline where we've gone all the way from raw reads to figuring out which reads are probably real and what their frequencies are, and assigning taxonomies to them, even potentially down to the species level. So I'll stick around for a few minutes in case anybody has questions. Feel free to jump in with audio, or feel free to jump in with chat. And if there aren't any questions after a few minutes, I will end the meeting for everyone. Yes. So so Vivian is asking, where are these taxonomic assignments coming from? Are they being pulled from a reference? And the answer to that is yes. They're. You'll notice that in your data file you have 4 files ending in FASTQ, and those are obviously reads. You also have another set of files in there that involve the name Silva, and that's using the Silva database of ribosomal RNA sequences. So that's actually the database that's tying together ribosomal RNA sequences with what species they originate from. Okay, so I have a question on here.
Sorry, that second one, I didn't copy paste the rest of it.
Ah.
Oh, you know what? Is it because I don't have, I see it.
Oh, okay.
I didn't have a quotation, I think.
Michael?
Yes.
I just had one kind of general question. Obviously, I noticed you're using R, and I have been really used to using Python and using like my Conda environments for that. Do you suggest using R or Python for different types of analyses that you're running? Does one kind of work faster than another, or?
Actually, my preference is Python.
Okay. Okay.
So actually, a lot of what you're seeing here, I actually have packages I made for automating bioinformatics analysis. And for example, I wrote a package that will automatically analyze microbiome standards for accuracy. And if you look inside that package, what you realize is it's running a Data2 pipeline to call taxonomy. But the way that it's running a Data2 pipeline to call taxonomy is it actually has Python running R.
Oh, cool.
So Python is actually generating R scripts on the fly, and then basically telling R to run the R scripts.
Wow.
R is actually not my main language. My main language is just head and shoulders Python. Like I can work in R okay, but Python's like basically my native language.
Yeah, I was a little worried that I needed to get a little more well-versed in ARBs, but so that makes me feel a little more happy.
Yes, no, there's actually some very good solutions now for people who run R who need to run Python programs from within R. There's actually solutions for them to be able to do that. And likewise, there's solutions now for having Python become able to run stuff in R.
Gotcha.
Occasionally, even Python can just directly control R.
Awesome. Very cool. Thank you.
Mm-hmm. All right, and I see somebody's getting an error message.
I guess I thought that was when I heard you say that we could put our own username in there. Am I incorrect in thinking that?
No, you should be able to put in your own username. That's fine.
Okay, then I don't know what that was coming from then, this error.
WS11. Oh, so it should be WS11/data/Silva whatever.
Am I missing the /data? Is that what it is? Okay.
Yep.
Thank you.
No problem.
And then when we quit out of R, it's asking to save the workspace. I'm assuming, is that yes, or does it, will we need this table and any of this stuff for tomorrow?
So basically right now, I think we're actually done with the computational portion.
Mm-hmm.
And actually, like I mentioned, you guys are the first class. So I'm going to polish up the computational portion. And I think for the future classes, I'm probably going to split it up over a couple days. So saving your workspace image is actually going to be important in the future, because I suspect in the future I'm probably going to divide this up over the 3 days of the course rather than having everybody run it at once.
Okay.
And I'm probably going to use, for example, the error modeling time. I'm probably going to put in. I'm probably going to talk about some topic during that time, because that error modeling takes a while. So that'll probably change a little bit. But yeah, for this one, we don't have to save our workspace.
Okay.
Because we're not going to go back to it tomorrow. Tomorrow, I'm going to basically kind of recap what we did and talk about some of the practical issues with microbiomes. So things like bioburden, sample collection issues, accuracy issues and biases, and that kind of thing.
Awesome. Okay, cool.
Yep.
Thank you so much. I'll see you tomorrow.
I'll see you tomorrow. All right, and if there's no further questions. All right, anybody with questions? Last chance.
How long should this taxa step or assigning the taxa take? Just out of curiosity.
Assigning taxa can take a few minutes. So if it takes like 5 minutes, it's not completely surprising.
Okay, great. Awesome. Well, then thank you so much.
Yep. All right. I'll talk to everyone later.